Hello, everyone. Thank you for joining us today. My name is Felicia Goodrum, and I'm the co-editor-in-chief for the Journal of Virology. The American Society for Microbiology, or ASM, publishes JVI, which is hosting today's webinar, as well as all others that we call the JVI Seminar Series. For more information on the series and to watch our introductory episodes, please visit the JVI website or scan the QR codes that you see on the slide here. Next slide, please. If you're not familiar with JVI, we publish preeminent work in virology research. We welcome primary research, mini reviews, and other article formats that investigate fundamental processes and structure in viruses, the mechanisms by which they interact and evolve with their host and environment, and novel methodologies, therapeutic, and diagnostic strategies. Next slide, please. Before I turn things over to our speakers, let me just quickly go through the format of this webinar. The first 35 minutes will be about the science, and that's where Adam Bailey will be presenting his work. This will be followed by a 10-minute discussion and Q&A session, and the remaining 15 minutes will be used to discuss how we can continue advocating for science. And we'll discuss questions such as why the study is important, what it means for your family and neighbors and the public, and we'll also ask our speaker to share his experience in going into science and his career as a scientist. Now for some housekeeping rules. The webinar and future JVI Seminar Series webinars will be recorded and posted on the JVI website. Our plan is to host these webinars every other month. In lieu of a certificate of attendance, you will receive an email confirming your registration for the webinar, and this will serve as your proof of attendance. We do not provide certificates of attendance for today's webinar. If you have any questions for our speakers, please use the Q&A button at the bottom of your Zoom screen. We will not be using the chat function, so submit all questions using the Q&A button. Our webinar today is entitled New Insights into Arterivirus Receptor Usage and Tropism. Our speaker will discuss his paper, Ineffectual Immunity in a Resurrected Mouse Model of Persistent Viremia. This was an editor's pick and was published in JVI in May of 2025. If you have not read his paper, I highly encourage you to do so. It's open access, and so it's free and available on the JVI website. Next slide. Now I would like to introduce one of our fabulous editors, Dr. David Levy, to introduce our topic and our speaker today. David?
Hello, everybody. Thanks for joining us. I'm David Levy. I'm a professor at New York University School of Medicine here in New York City in the Department of Pathology and in the Department of Microbiology, and also an editor at JVI. I largely handle mini reviews and what we call gems, which are short pieces about emerging topics in virology, but also occasionally handle basic research papers, such as the one we'll be discussing today, which came across my desk earlier this year, and I was really intrigued by it because this paper addresses, I think, a very important topic these days. You know, basically, we're all concerned about chronic disease, and we know that viruses play a fundamental role in many chronic diseases, particularly due to viral persistence. And persistence is one of the things we really don't understand that well. We have perhaps a better understanding of DNA viruses, how they persist, but for RNA viruses, particularly for positive strand RNA viruses, there's really a lot to understand. And one of the problems has been that there aren't really any good models to work with. And in this paper, Dr. Bailey has come up with a new system that could be used for looking at viral persistence with an RNA virus, one that seems to evade the immune system by mechanisms that we don't understand, and it persists for the life of the host without seeming to cause a lot of disease, but maybe there's things that are going on there that we really don't understand. So what I thought was really exciting about this sort of deceptively simple paper is that it introduces some really important concepts, probably raises more questions than it answers, and hopefully presents a way forward for people to pick up on this and do some fundamental research. So it's really great to have Adam Bailey here. Just briefly, Adam's been working in virology for many years since his undergraduate work at Colgate University. He started in virology. He has an MD and PhD degrees from the University of Wisconsin. He has worked on arteriviruses, which is what we'll be talking about today. He's also worked on other viral families. He did a medical fellowship in pathology and also a postdoctoral fellowship at the University of Washington University. And like many virologists, he got sidetracked into COVID during the pandemic and made some fundamental contributions to our understanding of that virus and how animal models can be used to better understand it and create vaccines. He's also done a lot of work on diagnostics in terms of providing new ways of diagnosing viruses. He's worked also on yellow fever virus, on pegiviruses, and on arteriviruses, which we'll be talking about here. He's trained with some of the luminaries in the field, like Jonathan Udell and Mike Diamond, and he's one of the up-and-coming virologists that we have now chosen as a new voice in virology, and he'll be publishing a mini review on receptor utilization by viruses sometime early next year. So it's really a pleasure to have Adam here and have him present his work.
Wow. Thank you, David, for that very generous introduction. And I want to thank you for this opportunity. It's a real honor. And thank you as well to Felicia and all of Journal of Virology. So today I'm going to tell you about my journey trying to understand viral persistence, but really sort of my journey has been mostly taking steps back so far to understand more fundamental aspects of viral biology, like receptor utilization and tropism, with the hopes of eventually connecting that to persistence. And so hopefully I can take you along that journey with me. So the first thing I want to tell you about is just how I became interested in these esoteric animal viruses known as arteriviruses. So early during my PhD in Dr. David O'Connor's laboratory at the University of Wisconsin-Madison, we essentially went hunting for novel viruses in wild African primates. And you can see some of the species that we obtained samples from here, and some of the sites all across sub-Saharan Africa. And the main goal of this virus hunting exercise was to identify novel simian immunodeficiency viruses, the relatives and sometimes ancestors to HIV. And indeed, we found many simian immunodeficiency viruses in these animals, but because the technology that we used was at the time relatively new, known as unbiased deep sequencing, this allowed us to look at all viruses that were at a certain level in the blood of these animals. And what really surprised us was the fact that many of them were infected with these viruses known as arteriviruses. At the time, only one monkey arterivirus had been described, and that was known as simian hemorrhagic fever virus. But we found these in a variety of different monkeys across sub-Saharan Africa, and some of these animals were actually co-infected with two highly divergent variants, this red colobus monkey here. So this raised a lot of questions for me right off the bat. But first, let me tell you a little bit about what are arteriviruses. So they're positive sense single-stranded RNA viruses with a lipid envelope. They're in the Arteriviridae family in the larger Nidovirales order, which also includes coronaviruses. The genetic diversity of these viruses is quite vast. The simian arteriviruses, many of which I helped discover, are shown here in yellow. But there are also arteriviruses that infect mice. There are arteriviruses that infect pigs, and also some that infect horses, among many other mammalian species. They have a whole range of different diseases and transmission modes that they can cause. So this ranges from everything from fatal hemorrhagic fever and encephalitis in some monkey species to chronic, persistent, subclinical infection in other monkeys. We really don't have any idea what determines whether or not a specific arterivirus host pairing will lead to disease, persistence, or even infection. The mouse arterivirus, which I'll talk about a little later today, is known as lactate dehydrogenase elevating virus. I don't like that name. I didn't come up with it, but I'm just going to call it LDV. And then the most well-known arterivirus is PRRS, or porcine reproductive and respiratory syndrome virus. This causes usually a mild respiratory infection in adult pigs, but is highly fatal for the fetus of an infected mother. And so farmers will wake up to what is horrifyingly called an abortion storm on their farm, where all of their pregnant sows have aborted. And so for this reason, PRRS causes billions of dollars in annual losses to the pig industry. And then there's also a horse arterivirus known as EAV. And I'll touch upon many of these viruses as I go through my talk. But essentially, they can transmit sexually, bloodborne, airborne, and many of them, though not all of them, appear to specifically infect macrophages. Interestingly, arteriviruses are currently the only family of mammalian RNA viruses that have never been shown to infect a human. So that also raises a lot of questions in terms of, well, is that just haven't infected a human yet? Or is there some intrinsic resistance that humans have that we just don't know about, that could be protecting us from these viruses? So I began to study these viruses in captive monkeys. And what you're seeing here are two different studies we did with two different arteriviruses. One is the classic simian hemorrhagic fever virus that was discovered in 1964 following an outbreak of hemorrhagic fever in a primate facility in Bethesda, Maryland, actually. And then another one is a virus that I discovered surreptitiously circulating in the Southwest National Primate Center. And as you can see, these viruses have extremely different phenotypes. These are all infected macaques, but the ones infected with simian hemorrhagic fever virus have very high viremia, rapidly succumb to the infection, whereas those infected with the Southwest baboon virus, that virus establishes a persistent viremia, and those animals are clinically fine. So this piqued my curiosity and really led to some fundamental questions that I'm still chasing today. How are these viruses able to persist? What determines whether an arterivirus will cause disease and or establish persistence? And this made me realize that the monkey is really a terrible host for investigating these questions for a variety of reasons. So that's basically where my PhD ended. And I took a 4-year break away from arteriviruses to study yellow fever virus and, like every other virologist on the planet, SARS-CoV-2. And then I started my own laboratory back at the University of Wisconsin-Madison in 2021. And I decided that I wanted to study arteriviruses, but I wanted to do it in a way that hadn't really been approached before. And so we have a two-pronged attack on arteriviruses. One is to study highly divergent family members in parallel to understand core attributes of this entire family, so that if one of these did emerge to infect people, God forbid, we would know something about it because we know some things about how the family as a whole behaves. And this has led to the identification of some novel arterivirus receptors that I'll tell you about shortly. And then in parallel, we've really been trying to resurrect the mouse arterivirus, LDV, as a research model to leverage the advantages of the murine host. And so this has led to the creation of the first cell culture systems for that virus, and also led us to a better understanding of tropism and transmission, which we are continuing to study. So one of the many features that makes arteriviruses unique and interesting is they have a very unusual glycoprotein topology on the surface of their virion. So unlike related coronaviruses, which essentially have one spike on their surface, it's encoded by one gene, arteriviruses have between 7 and 11 unique glycoproteins on their surface, and these coalesce into poorly defined complexes. So shown here is the minor complex, which is composed of GP2, 3, and 4. And then there's also a major complex composed of GP5 and M. The details of these is not pertinent to this talk, but I just wanted to highlight the fact that there are many different glycoproteins on the surface of this virion, and this is very unusual for an RNA virus. Interestingly, the simian arteriviruses have undergone an ancient gene duplication that has led to the expression of an additional 4 glycoprotein genes that have homology to the minor complex, and this is called prime complex. And we really don't understand exactly how many of these glycoproteins interact with receptors on the cell surface and in the endosome to mediate viral entry. What is known is that PERS, GP2 and GP4 interact with a cell surface molecule known as CD163. This is the hemoglobin haptoglobin scavenger receptor that is almost exclusively expressed on macrophages. And so this factor has been shown to be required for PERS infection. They've actually made CD163 knockout pigs that are completely resistant to infection. So it's clear that CD163 plays an important role in the viral entry process, at least for PERS. Other host factors are less well understood, and the host factors that the prime complex interacts with are completely unknown. So based on how many glycoproteins are on the virion surface and how few host factors are actually described, I hypothesized that there were more receptors to be discovered for this family. And so we found a cell line, the MA104 cell, which is an African green monkey kidney epithelial cell that supports replication of diverse arteriviruses. And you can see the cytopathic effect of SHFV, PERS, and EAV here in red, green, and blue, respectively, and the virus that is produced in the supernatant of these cultures on the right. And so we took advantage of a library, a genome-wide CRISPR knockout library that was made by the Broad Institute that actually targets the African green monkey genome. And so the way this worked is we introduced all of this library of guides in a pooled fashion into our MA104 cells. You can see the distribution of guide counts on the right here. And then we infected the cells, and then we sequenced the guides for this in the surviving cells to essentially look for host factors that were enriched that would provide some degree of protection from viral infection, such as a receptor. And these are the results. They're published, but one of the things that was really nice to see was that CD163 was a top hit for PERS and SHFV, but not interestingly, equine arteritis virus. If you're interested in learning more about what substitutes for CD163 in EAV infection, we recently published a paper in MBio on that, and the answer is CD81, which we think explains some of equine arteritis virus's expanded tropism relative to other arteriviruses. But one of the things that really jumped out at us right away was the identification of this gene FCGRT as a top hit for all 3, along with the gene B2M. So FCGRT encodes for this very interesting molecule that I've learned a lot about called the neonatal FC receptor. And the expression of the neonatal FC receptor, which I will call FcRn, on the surface of cells is dependent upon proper folding with the gene product of the B2M gene, which is beta-2 microglobulin. So those 2 hits from our screen are related because they complex together on the cell surface. And what this FcRn molecule does, it has many functions, but they all involve essentially transport of IgG molecules or antibodies across barriers in the cell. So in macrophages, when macrophages are penocytosing their environment, FcRn will bind to IgG and albumin in the acidified endosome, essentially rescuing those expensive molecules from lysosomal degradation. FcRn is also the primary molecule that transports maternal IgG across the placental barrier into the fetus, providing that passive immunity that we all learn about. So very interesting molecule, and now apparently a viral receptor. So we wanted to work that up further. So we then made single gene FcRn knockout MA104 cells. As you can see, those cells are completely protected from cytopathology when infected with SHFE, PERS, and equine arteritis virus. This is showing the production of infectious virus in the supernatant of those cultures, and the FcRn knockout cells are completely resistant to infection. We included SARS-CoV-2 here as an outlier that's in the same order but different family. So this does appear to be a family-wide effect, but not a order-wide feature of viral entry. Working with a collaborator at Ohio State, Cody Warren, he had recently shown that human ACHN cells that ectopically express human CD163 could be readily infected with simian hemorrhagic fever virus. However, if you knock out the neonatal Fc receptor in those cells, it also renders them completely resistant to infection, indicating that CD163 and FcRn are likely playing non-redundant roles in a cooperative entry process. So to then further hone in on entry as the step at which FcRn is playing a role in the viral life cycle, we transfected in viral genome into our FcRn knockout cells, and this led to production of infectious virus. So this essentially receptor bypass experiment is a classic one in virology, and so we show that here. We can also block infection with a monoclonal antibody that essentially covers up the neonatal Fc receptor, and so we can do that in a dose-dependent fashion here, again indicating that FcRn is involved in the entry step and can be blocked on the surface. We then additionally wanted to extend this finding to other cell types beyond MA104s. So what you're seeing here is a M-Cherry reporter simian hemorrhagic fever virus with and without antibody. So the anti-FcRn targeting antibody is orilenomab, which I will just call Ori, or an isotype control. And so you can see that cells that are exposed to our M-Cherry reporter virus turn red. Those that are given the isotype control stay red, but those that are given the anti-FcRn antibody are protected from infection. And we can do this in MA104 cells, the ACHN cells, ectopically expressing human CD163 that I mentioned before, and then also these macaque iPSC-derived macrophages. And this allowed us to then study some viruses that we can't yet get to grow on immortalized cell lines, such as some of those viruses that I discovered in jungle primates in Africa. And so on the left here, you're just seeing quantification of essentially that same experiment that I showed you on the previous slide. But the use of these iPSC macrophages from macaques has really been interesting because we've now been able to show that we can block infection of the Cabale red colobus virus and this other virus, PBJV, from an outbreak in Alamogordo, Texas from the 1980s, using the anti-FcRn monoclonal. One other thing that we discovered was that expression of FcRn ectopically really enhances the susceptibility of cells to arterivirus infection. So what you're seeing here is the dilution of the same stock, so the titration of the same exact virus stock on wild-type MA104 cells versus those that are ectopically expressing the neonatal FC receptor. And this alone leads to an increase in the quantification of the viral titer by about tenfold, suggesting that people who have historically been titering this virus and PERS on MA104 cells may be underestimating the amount of infectious virus in their stocks and samples. We can also express FcRn and CD163, in this case from the GRIVET. Each individually doesn't really do much in terms of rendering a non-permissive cell line like the Viro cell susceptible. However, if you express them together, you get massive CPE and large amounts of virus production, again, indicating that both of these things, neither of one is sufficient by themselves, but together they can make a cell as susceptible as a wild type MA104 cell. Finally, we also showed that different FcRn orthologs can function as species barriers in terms of cross-species infection. So I'll just point out one example here, but for example, the mouse FcRn ortholog can support EAV, but not PERS and not SHFV, indicating that, as we learn more about this, FcRn may be a molecule that is restricting or preventing infection of new species with these viruses. So conclusions for this part of the talk: FcRn is necessary but not sufficient for arterivirus infection of multiple cell lines. FcRn is co-opted by arteriviruses during the entry step of the viral life cycle. Its expression renders cells hypersusceptible to infection. And it acts as a receptor for every arterivirus that we've looked at to date. There's a lot of outstanding questions, and I can't do this all myself. So one thing we still want to know is, what is the precise role of FcRn in the entry process? Is this a stepwise thing, or do FcRn and CD163 bind together at the same time? How is this coordinated? And then from a physiological perspective, FcRn is very important to maintaining circulating levels of IgG. And so are these viruses antagonizing this process as a way to sort of circumvent humoral immunity? These viruses are also very efficiently transmitted vertically. And so is FcRn playing a role in that? These are questions that we are gearing up to answer, but have not yet had the chance to tackle. So in the last part of the talk here, I'm just going to tell you about some of the work we've been doing to resurrect LDV as a research tool for understanding viral persistence. So this is a viral load trajectory of LDV. It amazingly goes to 10 to the 10th genome copies per mil of serum within 24 hours of an infected mouse. These mice are fine. And then over the next 60 days, the viral load will come down to a set point of about 1 million genome copies per mil, and it stays that way for as long as we can tell. We've looked out to 400 days, but presumably for life. So this virus was discovered kind of as like an accident, basically a contaminant of some tumor models back in the 1960s. It was initially studied as a model of failed immunity or persistence, but nobody could grow it in immortalized cell lines. And so as more and more tools came out for its sort of competitor, lymphocytic choriomeningitis virus, or LCMV, you can see that LCMV really takes off in the 80s and 90s, and LDV kind of falls by the wayside as a consequence. So we're interested in understanding how this virus is able to persist. And one of the things that we've found is useful in measuring is when you look at the viral load trajectory of many persistent viruses, HIV, hepatitis C, many of these viruses exhibit an acute phase peak of infection, and then they come down to a set point. And LDV does this exact same thing. And so one question we've been asking is, what determines that drop in viremia from peak to set point? We've been working on another model of persistent infection known as mouse-adapted Pegivirus, which I don't have time to get into today, but this is published. And what you're seeing here is that compared to wild-type mice, which again exhibit that acute phase peak and then decrease to set point, animals that lack an adaptive immune system, in other words, animals that have RAG knocked out, they are unable to, they don't exhibit this peak to set point dichotomy. Their viral loads go high and they stay high, indicating that the adaptive immune system really is responsible for driving that viral load down to the set point. So we wondered, is something similar happening with LDV? And because we can now do this in the murine host, we put LDV through a panel of different knockout mice. And we were really astounded to see that basically it doesn't make a difference. You can put this virus into interferon alpha knockout mice, RAG deficient mice, and yes, there's some minor differences early in the course of infection, but by 100 days post-infection, the viral loads are all statistically the same. And so this really indicates that, in fact, the immune system is not driving viral loads down from an acute phase peak to a set point. And so what else could explain this? And right now, the only thing that seems to make sense to us is the fact that there are only a certain amount of target cells that are available during in the naive host that then get obliterated and become non-susceptible into the chronic phase of infection. And so this also suggests that immune responses are highly ineffective at controlling LDV replication. One thing we also wanted to know, based on the LCMV literature, was, does LDV induce tolerance similar to LCMV? So tolerance essentially puts the brakes on the immune response, and there's a variety of reasons for why the immune system has evolved a check on itself like this. But in LCMV, if you put LCMV into a PD-1 knockout mouse, and PD-1 is the master regulator of tolerance, those mice will die of immunopathology within 2 weeks. Essentially, the immune system goes crazy trying to clear the infection, and those mice will die not from the infection, but from the resulting immunopathology. So we put LDV into PD-1 knockout mice, and what we saw was, again, very surprising. The mice are completely fine in terms of their weight gain over time, but it also has no impact on the viral load trajectory, indicating that tolerance just isn't playing a role in persistence of this virus. So how does this work? Well, we need better tools to study this virus in this host. And so we, again, are trying to focus on understanding the tropism of this virus, because it appears that, based on our current hypothesis at least, the availability of target cells really is what's dictating the viremia curve in this model. And we need ways of manipulating the virus. So based on CD163 playing a role for entry in other arteriviruses, we essentially did a simple experiment where we looked at different macrophage cell lines from mice that are not susceptible. So you see that here on the left, compared to peritoneal macrophages, which are highly susceptible. And then we screened them for expression of CD163, and lo and behold, all of the murine macrophage cell lines have somehow lost CD163 when they became immortalized. And so when we ectopically express mouse CD163 on basically any cell line, we can render it highly permissive to LDV. And so you're seeing that here, where CD163 expression is shown by flow cytometry, and then the resulting PFU production and and gene copy production in the supernatant of those cultures. This is a really grainy photo of a plaque assay, but I like to show it because it's the first plaque assay ever for LDV. So we've optimized it a little since then and taken better pictures, but I always think it's kind of fun to show that. Then, of course, with the knockout mice, we can show that CD163 is indeed required for in vivo infection. And so now we have a better understanding of the molecules involved. We have some cell culture systems. And so we've kind of taken it from an in vivo finding, making it into an in vitro tool. And now we're making all sorts of recombinant LDVs that can teach us things beyond what we already know for the in vivo infection. So I'll just end by kind of showing that we've established that, yes, like all the other arteriviruses, LDV also uses FcRn to enter cells. And so you can see that here with increasing concentrations of lanalimab and our M-Cherry LDV reporter virus. We can show this on primary peritoneal macrophages as well. And one of the things that we've been doing recently that's been really exciting is we created a nanoluciferase expressing LDV. And watching this thing move around the body over the course of infection has been pretty wild. So this is a imaging of a mouse at 1 day post inoculation. And usually when we collect tissues to study where viruses infect, we just trim off the fat. But this really clued us into the fact that actually this virus is infecting macrophages in the adipose tissue as part of its acute phase burst. And so we never would have really looked there if not for this sort of approach. And so this is a little bit of a teaser. We're working on developing this further, and hopefully we'll have a manuscript out on this soon. But with that, I just want to thank my team, particularly a talented technician, Tressa Shaw, has really pushed a lot of this technology forward. Sarah Maloney did a lot of the EAV CD81 work that I didn't have time to talk about. And Logan Borsinger has really pushed forward the nanoluciferase and imaging studies. So with that, I'd be happy to take any questions, and thank you for your time.
Great, thank you, Adam. That was really a whirlwind tour of these viruses and very exciting work. Yeah, if we open for questions, we have a question and answer box in Zoom that you can type in your questions if you want. Maybe I kick things off a little bit here. I mean, you still have this issue of the virus goes from acute to a set point. Doesn't seem to be RAG2 dependent, but it's got to be dependent on something. Do you think it's just changes in the cells that are infected, that there's acute cells that are initially infected and then they're destroyed, but then there's other cells that can be infected, but for some reason are resistant to the cytopathology?
Yeah, so I think that's certainly a possibility. Our current hypothesis has a few tweaks on that working understanding. So um, there's a couple things that I think are worth mentioning. One is that most people seem to think that this virus is always cytopathic, but there's probably some pool of premature macrophage precursors that don't express CD163, for example, that create a renewable resource that can be continually infected. One thing we've been following up on in terms of the adipose story, at least, is so we can see the adipose resident macrophages get infected and they get wiped out. But then by about day 5, they start repopulating the adipose. But for some reason, they have learned to turn off CD163 expression. So they cannot be infected, but they are still there as macrophages. And so how that affects their function and how they sort of like know to do this is kind of an interesting twist on um, you know, genetic immunity in some ways. So we're working on understanding that further.
And is there an immune response to the virus? Can you see
So,
your T cell or B cell?
unlike our other model, viruses, in which we really can't detect a significant immune response, at least not a canonical immune response, there's definitely an immune response. It just seems to be highly ineffective. And so, um, we've done some of these more of these studies with the simian arteriviruses, but you can see the antibody attack and escape happen almost in real time. These viruses just throw up many different what has been dubbed in the PERS field decoy epitopes that essentially their whole purpose is to be highly immunogenic, attract antibody responses just so that they can then mutate away from them. Um, but when you think about our current arsenal of countermeasures for emerging viruses, they're pretty dependent on antibody responses. Monoclonal antibodies have had some success in treating viruses like SARS-CoV-2, but the biggest correlate of vaccine-induced immune protection is antibodies. And so these viruses appear to be poised to circumvent some of our best weapons, which is what makes me nervous from a pandemic preparedness perspective.
Right, so that, there's a couple of questions coming into the chat. I guess people are fascinated, why is there not a human virus? What are your thoughts about what prevents there from being a human virus? Is there genetic heterogeneity in any of the replicase genes that would limit the host specificity of the virus, or other aspects of the virus, that are preventing this from actually becoming a human virus?
Yeah. So one of the big challenges is that um, we have we don't even have isolates for the vast majority of the simian arteriviruses, and there are probably many out there that we still haven't even discovered. And that's just the simian ones, the ones that presumably have the lowest species barriers to infect a human. Um, this is where, looking at the different orthologs and whether or not viruses can use those for entry can be useful, but that's only one part of the equation. There can also be other incompatibilities beyond entry and or restriction factors that can shut down a nascent infection. Um, this is all like very speculative and preliminary at this point. We really have the research just needs to be done, but hasn't been done. Um, one thing that is clear is that humans have been and probably continue to be exposed to these viruses through practices like bushmeat hunting, et cetera. And whether or not other great apes like chimpanzees that regularly hunt red colobus monkeys are infected with these viruses, like it's just a big unknown.
And what are your thoughts on so in some cases you get really severe disease with these viruses. In other cases you get this persistence and there's no disease. So what are the differences going on there? There's a question about encephalitis caused by simian arteriviruses. What are the pathogenic versus non-pathogenic mechanisms that might be going on here?
That also is a great question, with really not well characterized answer. My hypothesis is that um, different suites of receptor utilization will allow these viruses access to different macrophage compartments. And those different macrophage compartments may be prone to initiating a anti-inflammatory or inflammatory response to the infection, because there's many viruses that infect macrophages that do really nasty things. And so, maybe FcRn is just one component of that. CD163 is obviously very important, but there's possibly other co-receptors that, if a virus can use it or not, can dictate slight changes in tropism that ultimately percolate up to different cytokine responses, different immunopathology, and ultimately manifest as disease. But this is just speculation at this point.
There's also a question here about, are there defective viral genomes created by these viruses, since that's often with other viral species an issue in persistence. So if you detect it in your deep sequencing discovery mode, partial viruses, truncated viruses that might be playing a role in persistence.
Um, I've not spent a lot of time looking at that, and it's probably something that someone should do. There's just so much low-hanging fruit to pick that, one of the questions we have in lab meeting is like, okay, we could do this, but should we do it? Because it comes at an opportunity cost, right? And so the defective viral genomes has just not risen to the threshold of like, okay, yeah, let's look at that.
So I thought it was interesting that beta-2 microglobulin is a component in entry. And of course, that's also involved in MHC expression on the cell surface. And there's other viruses that target that as a mechanism of persistence. Is there, does the virus actually directly targeting beta-2M to prevent something from going on other than viral entry?
It's a good question, and we have somewhat neglected investigating B2M at the expense of FcRn, and my hope is I can return to that and kind of flesh that out in a little more detail. Right now, we're trying to map the specific interaction between what is still an unknown viral glycoprotein or proteins, and the neonatal Fc receptor. But it is totally possible that B2M is playing a role in that interaction. I've just been learning a lot about this neonatal Fc receptor molecule, and it's fascinating all the things that it does. But it was news to me as well that B2M complexes with other things besides MHC, and it also is complexes with HFE, which is the gene involved in hemochromatosis. So it actually can stabilize and complex with a couple different molecules on the surface of the cell, which I didn't know until I started reading about this.
So one other question that comes up, many viruses that escape, particularly innate immunity, encode effector molecules that particularly inactivate the innate immune response, and that's how they get around it. Is there any evidence for that in arteriviruses?
There is some evidence for that in mainly it's been looked at in PERS. And there is some evidence that, kind of the usual suspects in terms of interferon signaling and things like that are antagonized to a degree. Much like coronaviruses, arteriviruses express a very large polyprotein of non-structural proteins that then get proteolytically processed into different subunits. And so many of those immune antagonism functions, at least at the innate immune level, are encoded by those non-structural genes.
Great. I just want to thank Adam and David for a fantastic discussion. This has been really wonderful. And we'll move into the science advocacy portion of our discussion. So in this portion of our JVI seminar series, we discussed the importance of the science. I would like to welcome Adam Bailey. He's given a great presentation, and our JVI editor, David Levy. Adam is a professor at the University of Wisconsin in Madison, and he's just talked about resurrecting a little-known virus that can be used to study persistence. So Adam, thanks for being here. And if you could just give us a brief overview of some of your findings and tell us exactly why ordinary people should be interested in the work that you're doing.
Yeah, well, thanks for having me. It's been really fun. So my lab over the last 4 years, and then when I was a PhD student before that, studies these little known animal viruses known as arteriviruses, and they have many interesting biological features. And so we're interested in understanding how these viruses work. But at the whole organism level, one of the most interesting features of their biology is their ability to infect an animal for life. And this uh, is unusual for an RNA virus, but it's even more unusual for a virus to do this in a way that causes very high levels of virus in the blood, like this virus does. And so we're interested in understanding how the virus does this for a variety of reasons, but I think, one of the most compelling and easy to understand reasons is this virus has figured out how to trick the immune system. And we, as scientists and clinicians, um, want to manipulate the immune system for a variety of reasons, like making better vaccines or treating cancer. And so, using this as a probe, if you will, to understand how the immune system essentially flunks when trying to control this infection, I think can teach us new things about the immune system. And so part of what I just presented on is about that.
I couldn't agree more. I mean, viral persistence is something that we understand so little about, from RNA viruses now to viruses that we've understand persistence for a really long time, like the herpes viruses. And these mechanisms are really important to controlling disease. So thank you for that work. David, do you have any thoughts or questions on the persistence aspect before we sort of talk to Adam about his career path? And
It's interesting how much macrophages are a target of many different viruses. And, particularly since sort of the job of macrophages is defense. So do you have any thoughts on, how this interplay, can be better investigated and maybe exploited as a way of preventing, treating persistence by somehow getting macrophages to do better at what they're supposed to be doing rather than replicating viruses? And I guess one other question, one of the things that really comes to mind here is, we have animal species like bats that seem to be able to have persistence of many different viruses without showing disease. And so, are we seeing another issue here where we have, um, you know, a persistence in an animal population that could be percolating and then causing problems down the road some way with maybe just a small mutation that then allows it to emerge as a human pandemic virus?
well, that's a great point, David.
There's a lot to unpack there. I think one of the things that I'll kind of work backwards, but one of the things that I like to think about is what viral infections look like in their natural host. So, for many viruses that uh, humans have, take SARS-CoV-2 as the most recent example. That virus has been circulating in a wild animal population for a long, long time, and it's had lots of time to co-evolve with that host to essentially maximize transmission, which often involves reducing disease. But when it's introduced into humans for the first time, that evolutionary negotiation hasn't really had time to take place. And we're kind of seeing it play out in real time now as the virus gets, less and less pathogenic over time. And there's many factors that contribute to that. But these viruses clearly seem to also fit that description of not really causing much disease in their natural host, But then when they emerge in a new host, for example, like PRRS in the pigs, which that virus came onto the scene in the 1980s. We still don't know where it came from, but possibly a rodent host, given that it's closely related to our mouse arterivirus. And anyways, I think it's interesting to recognize that persistence has as much to do with the virus as it does with the host. And it's really sort of that specific interplay between virus and host that can lead to the persistence phenotype. Talking about macrophages, I think it is interesting that many viruses seem to target macrophages and their main role is in defense. But most, macrophage defense is thought to be against larger extracellular organisms like bacteria. And so, one thing that may make them more susceptible to these infections is the fact that they're constantly sampling their environment and taking in non-specifically large amounts of fluid from their environment. And this may provide a unique Achilles heel that viruses can exploit to infect. One thing that's been, I resurrected the mouse arterivirus model because there are so many great genetic tools and resources available for studying the immune system in mice. But one thing that's actually been surprising is the lack of tools to study macrophages in mice. And so I think that's an area where we are hoping to push things forward, and then also working with other macrophage biologists, really start to understand, like, well, why are some macrophages susceptible to this infection and others are not, and how does that impact persistence and disease? These are sort of big picture questions that we really want to answer. And right now, the tools don't really exist to answer those questions with the degree of specificity we want.
I couldn't agree with you more there. I mean, have put so much focus on B cells and T cells as your major immune fighters. The macrophages have really lagged behind, and for persistent viruses, including the herpes viruses, I mean, they're playing a huge role. And we know even less about their role of these immune cells in tissues, which is actually where they're really doing their work. And I think where viruses are finding the safe harbor in macrophages. So I look forward to that. Adam, why don't you tell us a little bit about your career journey in science and what brought you to these questions, and really what fascinates you about being a scientist?
Yeah, thanks for asking. So I have always been a germaphobe, which is a weird thing for a virologist to say, but it's kind of like a moth to the flame situation for me. I wanted to challenge myself and see if my fear of germs was real and overcomable, and the answer to both of those is, I guess, like sort of. Um, so I initially I had always been reading about viruses, and a book that I read that was really impactful for me was The Great Influenza by David Barry, and it describes the 1918 influenza pandemic in a lot of detail. But the thing that struck me was that there's no reason this couldn't happen today. And so I started, working part-time in a virus lab in undergrad, and slowly kind of got more and more interested. And I thought I had always wanted to go to medical school, but then after a few of my research experiences, I decided that I just couldn't decide. So that's why I did the MD PhD program. Um, and then I always thought I wanted to be an infectious disease doctor, but it turns out that that was a little too much for my germophobia. And so instead I became a pathologist. And so I still am heavily involved in microbiology. I help run the microbiology lab at UW Hospital. But it's in a very contained setting, as opposed to, patients who can potentially be infectious and Probably the best thing for my germophobia has been having children because you just can't escape
They're
it in many
incubators.
ways. So, so all of these things have kind of coalesced around an interest in viruses and continuing to study how viruses cause disease. And then I think it's very interesting to juxtapose that with how viruses don't cause disease. And so we have a very active yellow fever virus research program where we work closely with physicians in Brazil using a hamster model to understand how yellow fever kills people. But then we also have at the other end of the spectrum these um, mouse viruses like arteriviruses and pegiviruses that appear to cause no disease. And so, how can both of those things be true at the same time is something that keeps me going.
And I think, Adam, you need to advance your slides. Sorry, I was just
Sorry.
noticing that. So we're sort of in the science advocacy part. Thank you. So one last question with regards to that, given your interest in human disease and medicine, can you talk a little bit about the importance of using an animal model for studies in virology and why is that important and what are the benefits there?
This is an important question and a hot topic right now. I think as we continue to refine our non-animal systems like organoids and artificial intelligence, the goal is always to use animals as little as possible and obviously as humanely as possible. But when studying really complex systems like the immune system, or when trying to understand the cells that a virus infects in the body, there's simply no replacement for animal model studies. And so I think nobody likes experimenting on animals, but people do like having new drugs and new therapies. And the alternative is either we don't develop new drugs and new therapies and make new discoveries using animal model research, or we have humans be the first test subject. So I think the public has sort of gotten into the habit of having their cake and eating it too on this topic. And I'm a big proponent of animal model research, not because I like experimenting on animals, but because the questions that really motivate me are these understanding these complex systems like the immune system, like the coagulation cascade, things that we just cannot simply model yet. And frankly, um, I don't think we will ever be able to fully recapitulate some of the idiosyncrasies of these really complex systems. So that's why I do animal model research. Um,
And I think, one of the things that's so complicated, especially about studying persistence, is you have the biology of what's going on in infected cells, but then you also have this immune component and they go hand in hand. And it's really hard to understand the system without, understanding both pieces of it. And of course, there's many, many pieces. But yeah, so thank you for your work. This is
yeah.
really fantastic.
I've asked ChatGPT what cells support pegivirus infection, and it gets it wrong. So the animal still has some utility.
Very much so. Well, thank you, Adam and David, for leading this and choosing this paper. It was just fascinating work that I think there's so much more to come that's going to be really exciting. So next slide. So just some final reminders about JVI. Next slide. We really encourage your submissions. We want your manuscript. So please remember that JVI is interested in all sorts of science in the field of virology, from cutting technologies to computational analyses, studies on viral structure and assembly, genome replication, genetic diversity, all the virus-host interactions that we've been talking about today that result in a productive infection, or in some cases, viral persistence, cellular responses to infection, including the immune responses and how the virus modulates these. Viral vectors, vaccines, and antiviral agents are also of interest. So check out those QR codes there. Next slide. And please join us for our next seminar series webinar that will happen in January 2026. Very excited to have you join us then. Next slide. And if you have any questions, please don't hesitate to contact us. We are really excited always to engage with the JVI readership and have ideas on how to actually make JVI work better for not only virologists, but the greater scientific community and the greater public community in general. And in fact, having a virtual webinar series was a suggestion from a JVI reader and author. And so we took that into very serious consideration and launched this. So thank you both for your time today. Thank you everybody for joining us, and we'll look forward to seeing you next time.
Great. Thanks, Adam. Thanks, Felicia. This is
Thanks.
so much fun.
Thank you.